Students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about recombinant DNA technology. In my upcoming classes, we will discuss very important topics of plant biotechnology. So, one of the most important topic is recombinant DNA technology. So, in today's class, we will discuss this. So at first, what is rDNA or recombinant DNA technology? It is a laboratory process that creates new DNA molecule by joining DNA fragments from different sources often to introduce new traits or character. So basically rDNA technology or recombinant DNA technology creates a new combination of DNA. Generally DNA combination, so suppose this is a DNA combination. So this technology modifies the combination or set of genes or these nucleotides, okay, by combining or joining two DNA fragments from different sources to introduce a new character or to generate a desired product or protein. Generally, gene express itself by producing a protein. So our ultimate aim is to produce one protein, one desired product. So we have to combine two genes to produce a desired protein. Next is gene splicing. Uses enzyme like restriction enzyme and ligase to cut and paste DNA into vector, which is then introduced into host cell for cloning and expression of new genetic material. So at first, by using restriction enzyme, we have to cut our desired piece of gene. Next, we have to introduce it into a vector. So vector is basically like a vehicle through which we can introduce our desired piece of DNA inside a host cell. Inside the host cell, it will multiply and express itself, okay. And the application of this technology includes producing therapeutic proteins, important therapeutic proteins, developing genetically modified crops, and aiding diagnostic and forensics. So basically it helps humankind in many ways. So what is the basic procedure? What are the steps of rDNA technology? How to modify the DNA or gene? So at first, this is our DNA of interest or gene of interest. We have to cleave it and introduce in a plasmid. What is plasmid? Plasmid basically present in bacteria, which is the extra chromosomal DNA sequence of bacteria. So at first we have to cleave the plasmid and introduce our gene of interest to create a recombinant DNA. Then we have to introduce in a new bacterial cell, and inside that bacterial cell our desired product will be formed from that piece of gene or DNA. So what are the steps? At first, isolation of DNA. We have to isolate our desired gene or DNA, piece of DNA. Then cutting the DNA, restriction enzymes, generally restriction endonuclease, are used to cut both the gene of interest and the vector at specific site. Vectors are the vehicle through which we can introduce our gene of interest inside the bacterial cell. Next, joining of DNA. DNA ligase is used to join the isolated gene into the vector, forming a recombinant DNA molecule. Next is transformation. Then we have to introduce that piece of gene of interest with the vector inside the host cell by the process of transformation. Then amplification, the host cell replicate, multiply, making many copies of the recombinant DNA inside their body. A selectable marker is often included to ensure only cells containing recombinant DNA can survive. Next is expression. The host cell express the gene of interest producing desired protein. Next, we can extract that desired product, then purify it. Next, what are the tools used in this recombinant DNA technology? At first, restriction enzymes, which are called as molecular scissors, to cut DNA at specific recognition sequence. So molecular scissors cannot cut DNA at any point. It can cut in a specific recognition sequence. Next is DNA ligase, which act as molecular glue to seal the DNA fragments together. Next, vectors to carry that piece of DNA into the host cell. Okay. Next, what are the application of this technology? At first, in the field of medicine, to produce therapeutic proteins and other therapeutic products. Next, in agriculture, creation of genetically modified crops with enhanced characteristic features like pest resistance, stress resistance, then higher yield products. Next is industry, to produce enzymes and other biologically active compound for industrial use. Next is for in the field of research, for isolation and cloning of specific genes to study their function and structure. So these are the applications of recombinant DNA technology. So with this, I am concluding my lecture video here. I hope you liked it. So in this video, I have tried to give every points and details about recombinant DNA technology. So if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.